Hello, everybody, and welcome to our webinar on how to make your PSC quality control workflow more efficient. My name is Annick. I'm the marketing manager at STEM Genomics, and I'll just be introducing the topic today before passing the floor to Juline Vincent, our R&D Project Manager. Before we start, a few housekeeping rules. I hope you can hear me and see me okay. If it's difficult for you, you can refresh. You can go on Chrome. That's definitely the best navigation for this platform. And we're also going to put in the chat a link to a troubleshooting guide. So hopefully with all this, you'll be able to enjoy a good webinar experience. Okay, but let's get into today's topic. Scientists like yourselves working on pluripotent stem cells, when it comes to building a robust quality control workflow, have a series of norms and standards you can follow. One of them is the OECD guidance document on good in vitro method practices, which will help you with the regulatory use in human safety assessments. Another one is the ISO 24603, which specifies the requirements for the biobanking of human and mouse pluripotent stem cells. You can also refer to the European Commission Joint Center document, the guidance document on good cell and tissue culture practice. And last but not least, the ISSCR standards for human stem cell use in research, which will establish a minimum characterization and reporting criteria for you working with human stem cells in basic research. So all these documents and regulations are a great framework to use. However, scientists can find themselves overwhelmed trying to meet standards and do the right thing. You also under time pressures, budget constraints, and trying to meet those standards can sometimes mean multiplying vendors. And this obviously can stretch the budgets even further. So this is where STEM Genomics comes in. We've been working with stem cell scientists since 2018, and we have been developing innovative testing solutions using technologies like digital PCR or also NGS. We have been working from two laboratories, one in the US, in Durham, North Carolina, and another one in Montpellier in the south of France, helping scientists from all around the world, over 200 by now, from both academic and biotech backgrounds. And we can count 7 out of the 10 largest big pharmas amongst our clients. So hopefully, with all this experience we've acquired since the start of the company, we'll be able to share some interesting tips and learnings for you today on how to make your quality control workflow more efficient. So today's webinar is going to be in 2 parts. First, we're going to help you identify the important aspects and specificities of human pluripotent stem cells. The second part will be about selecting the right types of methods and technologies to incorporate in your QC. And we'll see each other again at the end of the session to go through some Q&As. So you have questions on the right-hand side of your screens. You can just send them to us during the presentation, and then we'll make sure we'll answer all of them, or we'll get back to you if we don't have time after the event. So without any further ado, let me introduce to you Juline Vincent, our R&D Project Manager, who's going to carry on this conversation.
Hello, thank you, Annick, for this kind introduction. And hello, everyone. Today, we are going to talk about the most important matters you need to pay attention to when you work with human pluripotent stem cells. So before diving into the subject of our normality, I have a question for you, and you can answer in the poll. In your opinion, how many passages does it take for an abnormality to take over your culture? So you can answer in the poll. And maybe I can give you the answer. So, as scientists specializing in pluripotent stem cells, I'm sure that you already know that iPS are prone to acquire abnormalities during their time in culture. This phenomenon has been described in several scientific publications as copy number variation. This kind of abnormality appears during stressful time like continuous passaging, reprogramming, or gene editing, and confers specific advantages to the cells. Such defects can invade the culture in only 5 to 10 passages. As mentioned by Dr. Marcela Garita Hernandez from the Ocular Genomics Institute of Harvard, it's very important to test your cell culture regularly. She said, "In my experience, you need to check your cell lines routinely, as nothing that I have found to date can help you predict genomic abnormalities without testing your cell lines regularly. If your raw material is not stable, it is very difficult to trust any differentiation changes down the line." Among these abnormalities, one is most significant. Do you know which one, and do you know its special features? Again, you can answer in the poll at the right of the screen. Again, I let you some few seconds before to answer. So, of course, I speak about the 20q abnormality. This abnormality counts for more than 20% of the most recurrent abnormalities. The BCL2L1 genes is involved in this defect. This gene encodes two protein isoforms, a pro-apoptotic isoform and an anti-apoptotic isoform. When there is a BCL2L1 amplification, the anti-apoptotic isoform is predominant and confers a growth advantage to the culture. Furthermore, the 20q abnormality is very small and it is not detected by the G-banding, which is the method of reference for the analysis of the genomic stability in hPSCs. So you may miss the most frequent abnormality of iPS by using the most recognized method. As René Mas from the Regenerative Medicine Center from Utrecht said, since establishing a QC strategy involving ICS digital PSC, we realized that the 20q appears very often. We really did not expect it to be present so frequently. This led us to implement ICS digital PSC every 10 passages. These abnormalities can be problematic, but the worst scenario is when you think you are working with a specific cell line that turns out to be a different one. So again, I have another question for you. In your opinion, what proportion of cell lines do you think are misidentified worldwide? Again, you can answer in the poll in the right part of the screen. Okay, so here we go. We can answer to the question together. So you know, this is not trivial when we see these figures, because according to the ICLAC, 1/5 to the 1/3 of the cell lines are misidentified worldwide. This issue is due to handling errors, mislabeling in labs, and inconsistent nomenclature in the literature. As we know, scientists help each other by publishing their work and their results and by sharing material. But sometimes this contributes to spread the problem. Without surprise, the HeLa lineage is the most widely used cell line in labs and also the most cross-contaminating. It is everywhere, even when you don't expect it. And more than 28 billion per year have been attributed to the use of misidentified cell lines. So it's a very huge amount. And the last problem, if I can say that, and finally the smallest but most vicious problem, the mycoplasma. In your opinion, what is the highest level of mycoplasma contamination worldwide? Again, you can answer in the poll. It's the last question for you. Okay, so if I can give you an answer, this map is very, very important because we show that mycoplasma is responsible for 35% of cell culture contamination worldwide, with rates reaching up to 60% in Japan. So it's very important. And this tiny bacteria is invisible through microscope, and it is not sensitive to antibiotics. And yet, in the event of mycoplasma contamination in a laboratory, it can take several months to eliminate the contamination and restore your cell line to good health. So now that we have an overview of the main matters to pay attention to when working with hPSCs, let's see which kind of quality control tests are suitable for most laboratories to be cost-effective. Again, according to our client Marcella, testing is the easy part of our work really. With all the solutions now available out there, scientists have no excuses for not testing. It's with this perspective that STEM Genomics has developed a range of tests to help scientists to manage these challenges easily. STEM Genomics offers 3 tests to assess the genetic integrity of hPSCs. 2 of them enable the analysis of structural and copy number abnormalities. The ICS Digital PSC, based on digital PCR technology, detects copy number variations. The duo ICS Karyo combines the precision of digital PCR and the completeness of the G-binding to detect structural abnormalities in addition to CNVs. The STEM-seq range is based on NGS. The STEM-seq panel detects 361 oncogenes, which are the most well-known genes associated with cancer. To achieve an exhaustive analysis, this analysis can be combined with the backbone of probes covering the whole genome and providing a sensitive CNV detection. This test is named the STEM-seq Plus. To complete your essential assay, you can check for the absence of mycoplasma contamination using the mycodigital test, and also test the identity of your cells using HTR. In working with hPSCs, their undifferentiated states and ability to differentiate into all somatic lineages should be demonstrated. So the pluridigital test is based on digital PCR again and provides insight into their status. STEM Genomics can therefore offer a range of essential tests for hPSCs quality control. All these tests have different turnaround time, going from 1 day for the digital PCR-based test to 5 weeks for NGS-based tests. But today, we would like to focus on the essential assays and how STEM Genomics developed a synergy between these 4 tests. I will introduce each of them to you. The first one, the ICS Digital, was developed to detect abnormalities as soon as possible and before they take over the culture. It detects the 28 most recurrent abnormalities with digital PCR. The sensitivity and high precision of this method make it possible to cover 93% of recurrent variants. Its multiplex capabilities and ease of use make it possible to deliver robust results in 1 to 3 days after sample reception. So in 3 days only, you can assess the genomic stability of your cell line. If you need more exhaustive results and want to see the smallest as well as the biggest abnormalities, digital PCR can be combined with G-banding thanks to the dual ICS karyo test. Structural rearrangement and smallest CNVs are both detected before critical steps, such as bank characterization, for example. Here is an example seen at STEM Genomics. A trisomy has been detected on the chromosome 12 by digital PCR and G-banding. However, only digital PCR detects the 20q abnormality, even though it is present at a very high frequency. So finally, G-banding and digital PCR complement each other very well. Once you are pretty sure of the genetic stability of your cells, you need to be sure of their identity. By using the well-known and recognized STR, short tandem repeat test, a unique genetic profile of your cells can be established. This profile can be compared at the beginning and at the end of the workflow to assess the identity and the absence of contaminants. 16 loci are analyzed, and up to 2% of contaminants can be detected to guarantee the identity of your cells. And finally, to complete the essential quality control, you can use the mycodigital test to the mycodigital test to assess the absence of mycoplasma in your cell line. This digital PCR test targets 60 species responsible for about 90 to 95% of infections. It is highly accurate with a sensitivity of 10 CFU per ml as recommended by Pharmacopeia. By using regularly our digital PCR tests, mycoplasma will not be able to establish itself in your cultures. And in order to make the workflow more efficient, STEM Genomics has created a synergy by offering the possibility of sending a single sample to perform all these tests. First, you must collect and ship your samples to STEM Genomics facilities in France or in the US. Then, our technical teams perform 1, 2, 3, even 3 tests and provides you with a fully interpreted reports for all tests within 3 business days after sample reception. Please just note that if you perform the dual ICS karyo test, the turnaround time will be at least 10 business days due to the G-banding tests. And now you may wonder, how to implement all these tests in your cell culture workflow. Here represents our vision of good quality control of your cell lines for research use only. The one-stop shop analysis covers all important QC steps, but then can be completed with more exhaustive tests. So upon receipt of your cells and at the end of the process, of course, we recommend using all tests to obtain a complete qualification of your source and your final material. During amplification and after clone screening, it's important to assess genomic stability, identity, and sterility. For clone screening, pluripotency must be checked too. For critical steps such as banking, we recommend the most exhaustive characterization to be sure of your backup cell lines. And after differentiation, we recommend using pluridigital tests to demonstrate hPSCs' ability to differentiate into all somatic lineages. And for this step, sterility and genomic stability must be tested too. So, to conclude, don't forget to take care of your cells and it will pay off. So thank you for your attention, and now we can go through the Q&A.
Thank you, Juline. Right, I can see a few questions in the that have been posted. So I'm actually going to start with Ulrich's question about the duo hPSC. We responded on the chat, but perhaps it's interesting for other people. So Ulrich was asking about the turnaround time for duo hPSC.
Yeah, so the turnaround time is from 10 days, 10 business days at the US. And in France, it's France and Europe, it's 20 business days.
And that is linked to the G-banding test.
The G-banding test is longer than just a digital PCR test. So we have to treat the cells, and it's longer. It's longer than a digital PCR.
Thank you. A question from Lina: are these methods qualified for GMP?
No. These methods are just for research use only for now, but many clients include our tests in GMP compliant workflows, so they validate our tests in their workflow.
Another question from Ulrich: how many cells are required to perform all 4 tests?
I said 1 billion, I think. Maybe, yeah. It's the same amount for the ICS digital test you already used, I think. We can with just 1 cell pellet, we can do all these tests. It's a big advantage of this solution.
Stephanie is asking us about the costs for all 4 tests.
For the costs, so the first important thing, of course, you have a discount if you buy all these tests together as the one-stop shop solution. But if you want a precise cost, our sales team will be pleased to provide you with a quote. So you can contact us in the chat, or maybe I can just send you the email address of our sales team, or if Patricia
we'll send you a follow-up email with the contact details of our sales team. So this type of questions will definitely follow up with you. So don't worry, Stephanie, we'll get back to you on this.
Yeah, the sales team get back to you.
A question from Stephen about STR. Is it possible to compare several STR profiles?
Yeah, we offer this possibility by you can compare several STR profile between your samples. If you have two samples you want to compare, we can do that. And yeah, it's possible. You just have to mention which cell lines must be compared with which one. So you just have to mention this when you send your samples. But yes, we can compare several profiles.
Another question about STR from Lina. Can you speak of the advantage of your STR assay compared to general STR assays?
I just want to say that our STR assay is, I think, is the same that other STR assay by technical aspects, but the biggest advantage of this STR is when you can perform several tests just by sending one samples. So yeah, we can perform this test easily and very quickly just with one sample, and we can add other tests as ICS digital based on digital PCR. So I think yes, it's the biggest advantage of our STR. It's to be combined with other tests specific to stem genomics.
Ulrich is asking a question about G-banding. I guess you need live cells for the G-banding.
Yeah, exactly. Good question. And of course, we need live cells for G-banding. So, and also we can perform G-banding from frozen cells. It's a very new service, but you can do that from frozen cells or live cells. And at STEM Genomics, our technical teams can culture your cells during a few days, and we collect cell pellets from the flasks, and we can perform the tests that I presented to you just a few minutes ago. So yeah, you can send live cells, and we can perform the 4 tests that that I talked about.
Laurie is asking if we will make other kits like ICS Digital PSC 24 prop test available.
For the moment, unfortunately, no. we just have the ICS Digital PSC 24 test or the 20Q only, but I think if you have the 24, the 20Q is included. So yeah, but we discuss internally to implement other tests, but yeah, I just can't say that.
We have a question from Narine. I'm actually going to answer that one, Juline. She's asking if it's possible for us to email all the guidelines we mentioned, the OECD, the ISO, etc. Narine, it's actually planned in the email follow-up from that webinar to send to all the people who are attending the webinar the link to the paper that was published in July this year, and basically sums up all these guidelines. I think they're extremely useful. So we had planned on sending you the whole paper and also all the references that we've used in this webinar. Another question, and that's probably the last question we will have time for. Crystal is asking if STR is better than fingerprinting.
Oh, I don't know the method of fingerprinting, so maybe I can search internally, and I can get back to you with an answer more accurate, because I don't know very well this technique, so I prefer to search more details about it to have an answer better than this.
Okay, well, I think we're just going to stop for now, but we've noted all the questions you sent. We've also noted some of you asked for quotes. We're going to follow up on this. Don't worry about that, and send you all the information you need. Thank you so much. Yes, you're going to have the recorded video. You're basically going to get the replay. as soon as we finished this recording. So thank you so much for your time today, for all the questions and all the participation. It's been very pleasant. Thank you so much. And
thank you,
what we're
everyone.
going to do now is just drop in the chat a link to a survey. So this way you can give us feedback on how you thought this webinar was, and that will help you for future events like these. So yeah, I'll just give you a few seconds to grab the link. And we're also going to send this link in email follow-up. Thank you very much, everybody, and have a good rest of the day.
Thank
Bye-bye.
you. Bye.